So my name is Michaela. I am one of the member of the Cardiff team working on GNE, and it's a very, it's a great pleasure to be here and share discussion on GNE and also to share the progress of the GNE project. And let me say that it's a double pleasure for me because I'm Sardinian, I'm from Cagliari. So when we started the collaboration with the University was, very important for me since I got my master degree here, I would say 24 years ago. Of course, it was fantastic to then work with all the members of the consortium from different universities. They are really expert in the field. Here is Cardiff University, is the main building. Although Fabrizio and myself were working at the School of Pharmacy, now we moved to the School of Chemistry. Fabrizio is the PI of this project at Cardiff, and James and Emma are working very hard on this project. James is a PhD student. He's working since now, I think, 1.5 years. Emma has just worked for 2 months at the synthesis and optimization of the prodrug doing her AMCAM project. So what we do, what is our expertise? We are organic chemists, medicinal chemists, you want to maybe think about, and our interest is translational drug discovery. Our main expertise before to start with carbohydrate was on nucleoside chemistry, phosphorus chemistry, and fluorine chemistry. And we have several projects now running in our laboratories at the School of Chemistry in Cardiff, which span from anti-cancer agent, antiviral, antibacterial, and also different rare disease. Which include CDG, include, of course, GNE, but also lysosomal storage disease. We have worked with several companies in partnership, especially, I would say, I need to mention UCANA, which was our sponsor for the anti-cancer project, which has led, as you're going to see later on in the presentation, to 3 drugs in clinical trial. So what we do? What we do for GNE? What is our expertise? Our expertise is to apply a prodrug technology. What does mean prodrug? So a prodrug is an active molecule that you give to a patient, and inside the body is transformed in the active species only when get to the target point. And of course require a specific design to that to be happening. So you can think about a drug which doesn't have maybe all the properties that you want. Maybe it's a potent drug in vitro on the cell, but it doesn't work in the body for several reasons. You want to think this drug to be linked to a masking unit, which you have to think is a chemical entity, nothing more. Is another bit of molecule attached to that. And when it's inside the body, in the right place, is activated and is releasing the drug that you want, the drug that has the effect, the therapeutic effect. So why you need to do that? Why a drug cannot, if it's potent in the cell, is not working? There are so many reasons why that can happen. Maybe as a poor solubility, maybe it's not absorbed properly, maybe it's toxic. There are so many reasons for which you want to maybe think about making a prodrug approach. I would say that in the case of GNE, the most important things were enhance the absorption of the drug. And I show you why we need that. So whenever we take a tablet, the tablet needs to follow several steps. First of all, it needs to dissolve in the gastrointestinal fluid. Then you need to move to the intestine. You need to survive to the environment in this compartment. Then it needs to be absorbed, go in the bloodstream, and then go to the liver. Everything that we take orally go to the liver and is metabolized. So it needs to survive to the liver, because in our case, we want the drug going to the muscle. And then go to the general circulation, go distributed, and finally arrive in the active site. So to have a compound like that, you need to have certain properties, not only efficacy. Otherwise, if it doesn't arrive in the target point, it doesn't have any therapeutic effect. And what are these properties? The properties are a balance between polar and non-polar. So what does it mean, polar and non-polar? So you have to think about a sugar. If we take some sugar, it will dissolve easily in water. But a sugar is not going to dissolve in oil. So that's what we call polarity and non-polarity. So what do we want? We want a polar molecule, we want a oil-like molecule. Well, it's difficult to say because to dissolve in the gastrointestinal fluid, you want something polar. But to be absorbed from the gut, you want something that is not polar. And again, once you get to the bloodstream, you want something that is enough polar because there is water there. But to cross the cell membrane, you want something non-polar. So you can now maybe understand the difficult to design something that has certain properties and survive to the metabolism in the body. So what we have done create this technology for nucleoside. And this was the invention of our former boss, Professor Chris Begigan at the Cardiff University, which has taught nucleoside, which are used as a chemotherapy and as antiviral, has a lot of inconvenience. They need a protein on the membrane to get inside. They can be destroyed by premature breakdown, by enzyme that we have in the body. And they need to be phosphorylated to arrive to the DNA, stop the cancer cell to replicate and kill the cancer or kill the virus. So what we can do to make the bioavailability and the absorption of this nucleoside better? He thought we can create a masking group on the phosphate moiety, which is very polar, which can enter the cell without need a transporter. It go by passive diffusion, so it just enter because it's like the membrane of the cell. Entering the cell, release the active moiety and release the monophosphate, which still need to be undergoing triphosphorylation. So you have to think something like that. This is your nucleoside analog, and this is your phosphate, and you mask this with two groups, which need not to be toxic, of course, and they are released in the body to release the active species. So phosphate cannot go in, but such a molecule with this masking group can go inside the cell membrane and be activated in the monophosphate and your non-toxic compound. Many different approach has been invented, and there are different methods in which this kind of chemical entities are activated. What was invented in our laboratories was the protide approach, which has an enzymatic activation. So if we look at a little bit more in detail on the structure, you have a drug, you have a phosphate, you have an aryloxy moiety, and you have an amino acid, a natural amino acid. And this is very important because when the drug is activated, the amino acid is not toxic. And also the aryloxy moiety is not toxic for the concentration that we are using. So you can use a lot of different amino acid, a different aryloxy moiety, and different ester. What are the advantages? Increase the membrane permeability, bypass the first phosphorylation, and resistant to catabolic enzyme. That has been proven with no nucleoside. That's the mechanism of activation for which the protide get inside the cell by passive diffusion and through different enzymatic processes activated. As a triphosphate interact with our polymerase or DNA and stop the replication of cancer cell and viral cell. So what we have been doing in all this program, antiviral, anti-cancer, we have been changing the different ester group, which we found very much important for the activation of the compound. We have changed the aryloxy moiety, which we have found very important for the lipophilicity of the compound. And we have also looked at the amino acid, a different amino acid, which can tune the stability, lipophilicity, and activation of the protide. So that has led to 3 blockbusters in the antiviral area. Unfortunately, not from our group, from a big pharmaceutical company, Gilead, which has taken 3 drugs, one for hepatitis C, one for HIV and HBV, and the last one, which was approved in an emergency situation for the COVID pandemic. So it's a solid technology that proved very, very efficient for antiviral. We have taken this in our laboratories forward for the anti-cancer. I was part of this program. Fabrizio was also part of this program. And 3 drugs from our laboratories made by us has reached the clinic. So the question was at this point, can we this peptide approach can be applied to anything that need to be phosphorylated to be an active compound, like for example, mannac phosphate. And I leave now the floor to Fabrizio, which will explain the application of this on carbohydrate.
Thank you very much. So can this technology be transferred to other molecules that need phosphorylation? Well, you probably know the answer already because we are all here. And the answer is yes. In a sense that here we have N-acetylglucosamine, and the project actually, Michaela started this project back in 2016 or down that day, because we can basically, everything that has this OH group here can be phosphorylated, can be modified with this technology. So this kind of all handle that you can attack this promoiety here. Okay, so as soon as you have this nice OH in there, you can functionalize your molecule and making a prodrugs. Okay, so in this program here, we start to see, because N-acetylglucosamine is really important when you have osteoarthritis. You have to take N-acetylglucosamine to restore your joint when you have osteoarthritis. But the problem is the mannac is really hard to get into the joint. You need to take a huge dose, 3-4 grams, in order to make this efficacy. Okay, and sometimes it's not the best drug that you can treat osteoarthritis, right? So when basically your cartilage, okay, you start, this is the healthy cartilage, okay, there is a protein, okay, there is a nice chain of protein, and what you see here are just sugar, okay, and when your cartilage is degrading, basically the cartilage lose all these sugars here. So what our body do is basically try to replace these sugar, glycosaminoglycans, okay, using basically, this is basically what happens when you have osteoarthritis. Okay, you have the degradation of these glycosaminoglycans. Okay, so the body tried to repair it, making more glucosaminoglycan using N-acetylglucosamine. So in the project, we say, okay, if we give glucosamine enter the path to the synthesis of glucosaminoglycans. Okay, but as you can see here, okay, we have N-acetylglucosamine 6-phosphate. So since then, okay, why instead of giving N-acetylglucosamine, we are going to be at the phosphate, which will be one step forward. Okay, so in principle, you bypass these steps here. And here is the technology that was used. It was, there was some positive results in this project, results in 2 nice papers. So you can always imagine that this strategy here can be applied to mannac as well, because there is basically short difference. And in 2017, we were approached by Cardiff Therapeutics, because the original had the idea in doing this, and they asked us to develop the chemistry in N-acetylmanosamine, which apparently they're very similar, but believe me, the chemistry is completely different. It took almost 1 year to develop this kind of chemistry I'm going to show you right now. So the idea is, okay, you give it mannac. Mannac is a very polar molecule. If I remember what Michaela told a few slides ago, okay, it's very polar, so it's very soluble in water. But when it's time to cross the cell membrane or something lipophilic, then we have trouble in there. Okay, it's very good for oral drugs, but when it's trying to cross the cell membrane, that is a difficult process. That's why we have transport that they can transfer the sugar within our body. Okay. So it needs to transport it, and here, as already I showed before, we have the first part of the sialic acid synthesis, the GNE. It doesn't work, this one. So on top, we have basically the N-acetylglucosamine, which is epimerized, is transformed into mannac. that is then phosphorylated. So if you give MANAC, you got this step here when you have the N-acetylmanosamine that can be phosphorylated and so on. But if the phosphorylation part of the enzyme is deficient, is not effective, then probably MANAC is not, will have difficult. Of course, there are, as Rudiger suggested, there are many other ways you can get the phosphorus in this position. So the idea was to make the prodrugs of the MANAC 6-phosphate. So this molecule here is now lipophilic, so it can cross the cell membrane, it can cross the gut membrane, so it can be absorbed easier, much easier. And by diffusion will get inside the cell, will be enzymatic cleaved, releasing MANAC 6-phosphate. So in this way, this is the hypothesis, you can bypass completely the GNE enzyme. So you don't care anymore if you have the epimerase or the kinase, they are not working because you bypass completely the enzyme. This is the hypothesis that we have in during this project here. We synthesize probably around this very small family of prodrugs, basically change the phenol here, the amino acid and the ester on the paracetylated sugar. And actually we just got lucky because we could test only 2 compounds after this in this program, because the company was bought by Cerecor and then this program was stopped. But one prodrug was showing promising results compared to the MANAC. You can see here, we measure the sialic acid in patient myoblast, and compared to MANAC, we were able to increase the level of sialic acid in this experiment. Often it was done in duplicate because there was not much money available, so it's only one prodrug, okay? But because this paper, we got in touch with Michaela, and then we developed in this project, we try to increase the SDR of these prodrugs, show, investigate more amino acid, more esters, and so on. So in basically what we did in this program, we have extended structure-activity relationship. Structure-activity relationship is probably self-explanatory. We have changed all the parts and see what is the activity, okay? And we try to understand if this molecule can be activated, because it's really important that this molecule must be activated enzymatically in order to release the phosphate, okay? And we also need to improve this chemistry for the scale-up, and if we have to make more prodrug of this for more study, we need to improve the synthesis. The first synthesis was not that efficient, but we managed to do that in this 3 years period. So what's next? Before saying what's next, I just would like to show this cartoon here, because this molecule here, when you synthesize this molecule, you have actually something that is more complex than the simple structure that I showed before. Just an analogy, if you look your left hand and right hands are mirror images, one of the other. Your hand is the same things, okay, but left-handed is different from right-handed, okay? The same can apply for your foot. Left foot is different from, in the space, this structure is different, but actually has the same things, okay? Our prodrugs are really made as a 4 stereoisomer. The same thing, but in the spatial different, you can see here, is a phosphoramidate the chemists love so much, okay? So we have this molecule here is the same structure, but the space disposition is different. So we have actually 4 different molecules, okay? What we call stereoisomers. When you try to develop a drug, especially nowadays, we need to basically investigate each single isomers. Okay, so we're actually working in trying to separate each of these isomers in order to test which one could be the best. Okay, we're currently working on these, and we basically managed to get 1 single isomers. Okay, we are trying to test it now. We were managed to get it. We don't need to synthesize 3 more of them, hopefully. Okay, because now FDA or EMI require the testing of the single isomers. It's very hard to develop a stereoisomeric drug, a mixture of stereoisomers. You need to investigate each single of them, okay? Or at least most of them, unless the synthesis is impossible to separate, okay? So finally, the next point is this: we need to conduct more structure-activity relationship, because what Michaela says before, we have a MANAC that is very soluble in water, okay? Our prodrug is very soluble in oil and probably is too much, Okay, so we need to refine the SAR in order to make the molecule even slightly more soluble in water than oil, in order to facilitate the delivery inside the cell. And so we need to compromise between lipophilicity and hydrophilicity in order to make a better drugs. Okay, so we can do this chemically, acting on the sugar, acting on the amino acid, and basically keep going with the SAR in the future stuff. Or we can use formulations. Okay, we have to use a formulation, and this is the topic of my next colleague. But before doing this, I just would like to thank Dr. Stephen Thomas, Patrick Crutcher. There was the two guys in the Korean that came to us with this project. Yana Sedlakova was really good master student, Erasmus student, who did a very good job, and all together with the other researcher, and James that is keep going with this research.